Bismillahirrahmanirrahim. Assalamu alaikum. This is Amanullah. You're watching my YouTube channel, Dr. Aman's video. Dear viewer, today we are going to discuss trypanosomiasis. Trypanosomiasis is a word used for the diseases caused by the different species of trypanosoma. There are three major species of trypanosoma which are human pathogen. These species include Trypnosoma Crozai, Trypnosoma gambiense, and Trypnosoma rhodesiensis. Before going to start detail of these species, I would like to request you to subscribe my YouTube channel Dr. Aman's video and also try to hit bell icon in order to get the notification for my upcoming video. So starting with Trypnosoma Crozai. Trypnosoma Crozai cause a disease which is known as Chagas disease, and this disease is also known as American trypanosomiasis. The life cycle of Trypnosoma crozai involve a vector which is known as rhidovid bugs. These rhidovid bugs actually act as a vector for the transmission of Trypnosoma crozai from one host to another host. This rhidovid bugs belongs to a genus Tritoma, and it is also known as cone nose or kissing bugs. Why it is known as cone nose or kissing bugs, we will discuss in the upcoming slide. And the humans and other animal act as a reservoir host for the Trypnosoma crozai. In short, we can say that Trypnosoma crozai complete its life cycle in two hosts. One is rhidovid bugs and the other is human or any other animal reservoir host. The animal reservoir host include domestic cats and dogs and also wild species such as armadillo, raccoon and rat. The cycle, before going to start the cycle of the rhidovid bugs, I would request my viewer to see the photos of these armadillo, raccoon and rat. I'm not going to paste these pictures or photos in my presentation because my presentation will not be eligible for the YouTube ads. Therefore, I request you to see these photos or picture on the Googles. So the life cycle of the rhidovid bugs or Trypnosoma crozai is somewhat like this. As we know that tritomine bug is the vector for the rhidovid for the Trypnosoma crozai. When this tritomine bugs takes a blood meal, what it does, it passes metacyclic trypomastigote into feces. Trypomastigote enter bite wounds or mucosal membrane such as the conjunctiva. So started with this stage 1, if a tritomine bug is infected with a Trypnosoma crozai, when this tritomine bugs bite on the uninfected human being, so this tritomine bugs take a blood meal and at the same time it pass feces or it defecate on the skin of the human being. This biting of the human being result in the itching or irritation of the skin. Therefore, human being cause rubbing of that skin, which result in the introduction of the metacyclic trypomastigate present in the feces of the tritomine bugs, because tritomine bugs pass feces when it bite on the human blood meal. Therefore, these feces are present near the bite site, and when human being rub his or her skin because of the irritation or itching, so these trypomastigode present in the feces get enter into the wound site. Then it entered into the human being, so the human stages start. Metacyclic trypomastigode penetrate various cell at bite wound site. Inside the cell they transform into a mastigode. When they are enter into the cell, inside the cell they are transformed into another stage which is known as a mastigode. A mastigode multiply by binary fission in the cell of the infected tissue. They can infect different tissue of the body including reticuloendothelial system cells, glial cells, or the cells of the heart or cardiac membrane. So these MST goat multiply inside the cell and intracellular mastigome transformed into trypomastigote, and then the cell burst out and the trypomastigote enter into the blood stream. These trypomastigote again infect other cell and transform into intracellular mastigote, and this cycle goes on into the human's tissue. But at the same time, when tritomine bugs takes a blood meal from the human being, so the trypomastigote present in the human bloods are ingested by the tritomine bugs in blood meal. These trypomastigote differentiate into epomastigote into midgut, and this epimastigote multiply into the midgut and transform into metacyclic trypomastigote into hindgut. These trypomastigote are again released into the feces of the tritomine bugs when it takes blood meal from the human beings. So the cycle is completed when the trypomastigote present in the feces enter into the site of the bite. So this is the life cycle of Trypnosoma Crozai, which completed into two hosts. One is human or any other animal reservoir, while the other is tritomine bugs. Now, what is the pathogenesis? As we know that tritomine bugs bite preferentially around the mouth or eyes, hence the name kissing bugs. As I told you in my previous slide, that the tritomine bugs or the tritomine bugs is also known as kissing bugs because it bite or it prefer to bite around the mouth or eyes. And I also told that it is also known as cone nose bugs because the shape of the head of the tritomine bugs is like a cone, therefore it is known as cone nose bugs. The mastigote can kill cells and cause inflammation, consisting mainly of mononuclear cell. Cardiac muscle is the most frequently and severely affected tissue, but other muscles or other tissue of the body can also be infected. In addition, neuronal damage lead to cardiac arrhythmias and loss of tone in the colon, which result in megacolon and megaesophagus. The neural the damage of the central nervous system result in the cardiac arrhythmia. Cardiac arrhythmia is a condition which results in the high or low rate of the heartbeat, and it also results in the loss of the tone of the colon. Tone is actually the tension in the muscles of the colon or esophagus. When these tensions are lost due to the neuronal damage, therefore the size of the colon or esophagus become larger, which is known as megacolon or megaesophagus. So during the acute phase, there are both trypomastigote into the blood and amastigote intracellular into the tissue. As we have discussed in the life cycle, the trypomastigotes are present freely in the blood of the human host, and amastigote are present intracellular into the tissue. In the chronic phase, the organism persists in the amastigote form only. Chagas disease has in the recipient of either blood transfusion or organ transplant from infected donor. It means that Trypnosoma crozai can be transmitted with blood transfusion or organ transplant, and the organism can also be transmitted congenitally from an infected mother to the fetus across the placenta. So these are the other mode of transmission besides the transmission of Trypnosoma crozai by the bite of rhidovid bugs. Clinical finding: the acute phase of Chagas disease consists of facial edema and a nodule near the bite, coupled with fever, lymphadenopathy, and hepatosplenomegaly. The acute phase resolves into 2 months, but most individuals then remain asymptomatic, but some progress to the chronic form with myocarditis and megacolon. As we have discussed, that it damage the tissue of the heart and also result in the loss of the tone of the esophagus and colon, which results in megacolon and megaesophagus. So death from chronic Chagas disease is usually due to the cardiac arrhythmias and cardiac failure. Laboratory diagnosis: acute disease is diagnosed by demonstrating the presence of trypanosomiasis in thick and thin film of the patient blood. As we know that trypanosomiasis is freely present into the blood of the patient. Both stain and wet preparation should be examined. The wet preparations is used for the motile organism because trypanosomiasis is a flagellated form and it is motile. Because the trypanosomiasis are not numerous in the blood, other diagnostic method may be required like stain preparation of a bone marrow aspirate or muscle biopsy specimen. But you must know that in muscle biopsy or bone marrow, we can see the amastigote stage, because inside the cell there is amastigote stage and in the blood there is a trypanosomiasis stage. So therefore, whenever you are going to examine the bone marrow or muscle biopsy, so you will have to look for amastigote inside the mononuclear cells. While when you are looking examining the blood film, then you will have to examine for the trypanosomiasis in the blood smear. These are the trypanosomiasis which can be seen into the blood smear. A typical trypanosomiasis shapes can be C shape, S shape or U shape. This is U shape trypanosomiasis. This is S shape trypanosomiasis, often seen in stained blood films. Appearance long and cylindrical, as here you can see. Nucleus is 1 in number, located anterior to the kinetoplast. It is the nucleus present into the trypanosomiasis of the rhedoid bugs. Other features: kinetoplast located in the posterior end. Here you can see in kinetoplast, or here you can see this is a kinetoplast which is present in the posterior end of the trypanosomiasis. Undulating membrane extending entire body length. Free flagellum extended from the interior end when present. This is a free flagellum which is extended from the interior end, while this is kinetoplast present in the posterior end. In this case, this is kinetoplast present in the posterior end, and this is a flagella which is extended from here to here from the interior end. So these are the trypanosomiasis which can be seen in the blood film of the infected patient. The amastigote of the Trypnosoma Crozai is similar to the amastigote of Leishmania, which is round to oval in shape, and nucleus is present off center and kinetoplast is present near to the nucleus. So you can see my lecture on Leishmania. Now trypnosoma, the two other species, Trypnosoma gambiense and Trypnosoma rhodescens, are diseases. These organisms cause sleeping sickness, which is known as African trypanosomiasis. They are also known as Trypnosoma brosai gambiense and Trypnosoma brosai rhodescens. These are the other names which you can see in literature or books. Important properties and life cycle. The morphology and life cycle of the two species are similar. The vector for both is the tsetse fly of genus Glossina, but different species of fly are involved for each species. Different species of Glossina are involved in the transmission of Trypnosoma gambiense and Trypnosoma rhodescens. It means that tsetse fly actually belong to the Glossina genera. So the different species of the Glossina genera are responsible for the transmission of Trypnosoma gambiense and Trypnosoma rhodescens. Human are the reservoir for gambiense, whereas Trypnosoma rhodescens has reservoir in both domestic animal and wild animal. The 3-week life cycle in the tsetse fly begin with the ingestion of trypanosomiasis in a blood meal from a reservoir. I will start with this stage 1. Tsetse fly take a blood meal, inject metacyclic trypanosomiasis. When tsetse fly taking blood meal, at the same time it inject the trypanosomiasis into the human skin, and then human stages begin. Injected metacyclic trypanosomiasis transformed into blood stream trypanosomiasis which are carried to other sites. So this metacyclic trypomastigote transformed into blood trypomastigote and the blood carries this trypomastigote to other sites. Trypomastigote multiply by binary fission in various body fluid, for example blood, lymph and spinal fluid. Trypomastigote and then trypomastigotes are freely present into the blood. So when CC fly bite on an infected human being, it ingests the trypomastigote present freely into the blood, and these bloodstream trypomastigote are ingested by CC fly. Then what happened? Bloodstream trypomastigote transformed into procyclic trypomastigote in CC fly. Midgut procyclic trypomastigote multiply by binary fission into the gut of the CC fly. Procyclic trypomastigote leave the midgut and transformed into epimastigote. Epimastigote multiply into salivary gland. They transform into metacyclic trypomastigote. And these metacyclic trypomastigotes are injected into the human being when CC fly bite for its next blood meal on the human being. Procyclic, metacyclic, these are the different stages of the trypomastigote. So Trypnosoma gabiense and Trypnosoma rhodescens pathogenesis. The trypomastigote spread from the skin through the blood to the lymph node and the brain. The trypomastigote from skins through the blood, as we know that these trypomastigotes are converted into blood trypomastigote, and the blood carries it to the different sites which include lymph node and the brain. The typical somnolence, sleeping sickness, progress to coma is a result of a demyelinating encephalitis. Myelin sheet is a protective sheet of the nerve fiber, so it is demyelinated due to the attack of the inflammatory attack of these two species. In the acute form, a cyclic fever spikes occur that is related to antigenic variation. It means that in acute phase there is a cyclic fever spikes. It means that fever occur and then it subside, then again there is a spike of fever and then it gets subside. It is due to the antigenic variation. The word antigenic variation mean that the Trypnosoma gambiense and rhodescens has the ability to continuously change the antigenic structure present on the surface of the parasites. So therefore, when immune system produced antibodies against one antigenic structure, and the immune system eliminate and neutralize that form of the Trypnosoma rhodescens and gambiense, then Trypnosoma produce another kind of antigen on the surface which are not recognized by the antibody. So again the spike of the fever begin. Then again immune system start producing antibody against that stage, that type of antigen. When immune system again neutralize and eliminate that form, then the fever gets subside. Then another type of antigens present on the surface of the parasites which again result in the spike of the fever. So this is the antigenic variation which occur in the Trypnosoma gambiense and Trypnosoma rhodescens. As antibody mediated agglutination and lysis of the trypomastigote occur, the fever subside. However, a few antigenic variants survive, multiply and cause a new fever spike. As I told you, this cycle repeats itself over a long period. The lytic antibody is directed against the surface glycoprotein. Clinical finding: although both species cause sleeping sickness, the progress of the disease differ. Gambiense induce disease run a low-grade chronic course over a few years, while rhodescens causes a more acute, rapidly progressive disease that, if untreated, is usually fatal within several months. Gambiense complete its course of infection in a few years, so it takes few years to complete to complete the course of the infection, while rhodescens complete its course of infection within a few months. So the initial lesion is an indurated skin ulcer at the site of the fly bite. Indurated skin ulcer mean that the hardness of the skin occur at the bite of the sites. After the organism enter the blood, intermittent weekly fever and lymphadenopathy develop because of the antigenic variation. There is intermittent weekly fever with spikes and down, spikes and down. Enlargement of the posterior cervical lymph node occur, which is known as winter bottom sign, is commonly seen in these infection of these two species. The encephalitis is characterized initially by headache, insomnia, loss of sleeping, and mood change, followed by muscle tremor, slurred speech, and apathy that progress to somnolence and coma. Untreated disease is usually fatal. During the early stages, microscopic examination of the blood, either wet films or thick or thin smear, revealed trypomastigote. The trypomastigote of the Trypnosoma gambiense and Redulus Trypanosoma rhodesiensis is similar to that of Trypnosoma Crozai. An aspirate of the chancre or enlarged lymph node can also demonstrate the parasites. It means that you can also take aspirate from the chancres produced at the bite site of the CC fly or enlarged lymph node, and these aspirates can also demonstrate the presence of the parasite. The presence of the trypnosome in the spinal fluid coupled with an elevated protein level and pleocytosis indicate that the patient has entered the late encephalitic stage. It means that when you detect the trypomastigote into spinal fluid, and also you see the elevated level of protein into the CSF and the increased number of the white blood cell in the CSF, this picture indicates that the patient has entered the late encephalitic stage. Including these laboratory diagnoses, you can also use serological tests, especially ELISA for NGM IgM antibody, which can be helpful in the diagnosis of trypanosoma infection. If you have any questions or suggestion, you can write me email on khonamtalvi@hotmail.com, or you can write me your question in comment box. Thank you so much for watching my YouTube channel. Fi Amanillah.